All right, so hi everyone, I'm Jen. I'm a graduate student in Roman Stalkers group, and in our lab we like to observe microbes interacting with their environment. So this movie here, sorry.
There you go.
I feel like I have two mics now, so hopefully it's okay. This movie here is of a marine bacterium growing across a nutrient-rich surface. And actually, studies of bacterial growth do something like this, or very similar in that they always observe bacteria growing in unchanging environments, which isn't really that realistic considering that in the wild, environments fluctuate. And this is clear when we zoom into the micron scale, the scale at which bacteria live. So here's a small 1 millimeter cube of water, and what I want you to see is first that it's really heterogeneous and that there are nutrient hotspots randomly scattered around this space. And so these sinking particles or cells are all exuding nutrient for bacteria to grow off of. And so what this world looks like for an individual bacterium as it swims from patch to patch is then a series of nutrient pulses such that it's experiencing the environment in a way in which the nutrient fluctuates around it. And so as I describe this project on bacterial growth in fluctuating environments, I would like to really also illustrate how considering these microscale interactions helps us think about these larger processes. So how does something so small impact something profoundly larger than itself? Minoshi just gave us a hint that if we consider the Earth's carbon cycle, where CO2 is entering the ocean and through photosynthesis, you have the conversion of it into organic carbon, which then microbes can eat and consume and grow off of. Turning this carbon into biomass allows that carbon to enter the main food chain and increases its likelihood of staying in the ocean and being sequestered there. Whereas if the carbon is not converted into biomass, then it's respired as CO2 and lost back to the atmosphere. So how does the microscale help us think about this? Well, if you imagine here a gazillion of those tiny little cubes that I've just showed you, the current literature averages across them. And so what that essentially does is assume growth in a constant nutrient environment, which is problematic because averaging ignores the temporal effects of nutrient availability. So I'll use an analogy here in that you and I are fairly fortunate in that we have these regular meal times, right, consistently. And so what if I took all the food you ate in a week and allocated it differently to you? Such that, say, maybe breakfast, lunch, and breakfast, lunch, and dinner has to happen all at once in a day, or if all the food in a week was shoved into a 1 hour period, or if maybe instead of just eating in chunks, you had to just incessantly sip at this really nasty dilute nutrient drink for the rest of your life. So yeah, imagine then, like if you think about bacteria in the environment, there are actually a huge variation of nutrient profiles that they might experience. So how do the concentrations of these and how does the time scale of these affect how microbes grow? And so it's interesting, I don't know the answer, but what I'll tell you is how I think these fluctuations matter. And it's in that bacteria coordinate their growth with nutrient availability. And so what I mean by that is cells will grow smaller and slower in nutrient-poor environments versus nutrient-rich ones. And if we look at the carbon budget of a cell or its energy budget, this is explained in that in nutrient-poor environments, say if the sole source of carbon is something like glucose, the cell then needs to convert this glucose into other things it needs to grow, like amino acids or nucleotides. And the enzymes that catalyze this process, they require energy to maintain, so that this maintenance energy detracts from the carbon that the cell would otherwise use to grow. And that in nutrient-rich environments that supply more of this material, that maintenance cost is less, allowing the cell then to shunt more carbon into biomass production. And so these diagrams I'm showing here are of what cells and their physiology would look like under stable growth conditions once they've adapted to either poor or rich nutrient environments. But how do cells then switch between these growth states? And what that looks like then is that there's this second cost, second kind of sink where carbon goes into, and it's this energy that's required to adapt physiologically. So then, thank you, if you then consider if fluctuations were to occur on the timescales at which bacteria do adapt to their environments, then this cost, this adaptation cost, ends up continually consuming carbon from the pool that bacteria can actually use to grow. And so the prediction there would be that fluctuations, actually, one more sentence, that bacteria growing in fluctuating environments might actually see decreased growth than in ones in constant ones. And so I'm running these experiments, and maybe you can ask me about them in my question time. Thanks.
Thank you very much. Question in the back.
Ruby.
Can you tell me about the experiments?
Yeah, thank you. And so I'm running these experiments in which I have two parallel conditions, one fluctuating and one not, and they deliver equal amounts of nutrient, such as here, just that one, right, is temporally delivered in chunks and one is not. So hopefully we'll be able to visualize cells growing and looking at how that differs.
Thank you very much.
Thanks.
And I'm going to hand the mic over to Jameson to explain that.